Great. Thank you so much for the kind introduction and for having me here today. It's a pleasure to talk to everybody. And it's funny that you bring up my weather prediction. I actually have a slide now where I talk about the parallels of modern high throughput biology to weather prediction and how it's finally come full circle. And I pulled it out of my talk last night in interest of time. So now you're making me regret that. But I'd be happy to talk to anybody about it offline. So briefly, just my disclosures that I'm on the scientific advisory board for Resistance Bio, and I'm a consultant for MESTAG and Merck. So, I think everyone in this room is really familiar with the multi-omics revolution and the single cell and spatial revolution and where those have come from. One of the things I'm a mathematician who's housed in a cancer center. So one of the things that we often think about is what is the translational or clinical relevance of what it is that we're learning from these high throughput data sets and high throughput biology. And so one of the things to keep in mind from a cancer perspective is that we're not only in a technologic revolution, we're also in a therapeutic revolution. Where around the same time that single cell technologies were coming up into vogue, immunotherapies were also really coming into their prime in cancer biology. And there's a wealth of a ton of different ways to target and harness the immune system in order to better attack cancers that have resulted in really unprecedented responses in cancers that were previously therapeutically untargeted. And so where does this put us that we're really at this sort of, convergent science approach where we have these high throughput technologies, we have these new therapies, and the question is, can we leverage them better in order to get better precision medicine approaches? And one of the things that we think about, if we think about the history of precision medicine and cancer, it's largely been genetic based, where the idea being if you know a single driver gene, you can figure out what's going on under the hood to figure out what gene you should target in the cancer cells. In the immune system, it gets a little bit more complicated where you have different cell types that really drive the phenotype. And then within each of those cells, you have different molecular states. So you have this multi-scale systems level approach within the tumor environment that's driving response and resistance to immunotherapy. And I would argue also to targeted therapies, but I'm not going to focus on that as much today. So we're really at this point now where we're primed of wanting to think, how can we use multi-omics approaches in a therapeutic context? So one thing that's sort of the canonical approach to cancer immunotherapy and how people think about immunotherapy biomarkers is that you have immunotherapy responsive tumors. And these are tumors that are thought to have a large number of antigens that are presented on the tumor cells. T cells are able to infiltrate those tumors, come in, recognize the foreign antigen that's presented on the tumor cells, and go in and kill them. And often this is just achieved through activation with an immune checkpoint inhibitor that's designed to block the mechanisms that are telling T cells not to attack. So it's all primed to be there. In immunosuppressive tumors that are not responsive to immunotherapy, you could turn on those T cells as much as you want, but what happens is that there's a large number of immunosuppressive cells. So these are cells that block the T cell response, and so they don't allow T cells to infiltrate. And so even if you've turned on these hyperactivated tumor cells or T cells, they're not able to recognize the tumor cells and kill them. So this is right now one of the predominant biomarkers for cancer immunotherapy, along with tumor mutational burden that gives an indication of the antigens on the tumor cells. The challenge with this approach is that it takes a very binary view towards cell types, where it's thinking of, do you have T cells? Do you have immunosuppressive cells? And thinking of these as very binary features. One of the things that we've come to appreciate as a community in terms of computational biology and single cell technologies is that if you look at transcriptional data, there's a lot more there than just what cell type, what is your cellular identity, right? There's where are you phenotypically, and sort of that determines some of your function. Where are you along your evolutionary trajectory? Are you cycling? Where are you in cell state? and where are you spatially? And all of these different features are going to contribute to how a patient's going to respond to immunotherapy. And they're also going to moderate what we see in our single cell data, because we're going to see footprints of these different signatures that are independent of cell type. So then the question becomes, how do we learn these functional signatures independent of cell type from single cell data sets? And we're fortunate that this notion of having multiple sources adding up to give you different features of a data set is perfectly represented in the mathematics of matrix factorization. So I'm not going to go through my little cartoon of 4 genes that I often go through in more biologically oriented audiences. But suffice it to say, what we're thinking about is we have a world where we have gene expression in cells, say, from treated and untreated conditions. And we're going to use matrix factorization as a tool for latent factor analysis to figure out what are the patterns that are occurring, say, in the treated versus untreated groups, and then what genes are associated with each of those features that we see, so that we can uncover these underlying signatures in the data. So what Genevieve Stein O'Brien and my group realized was that we could adopt a Bayesian matrix factorization that we had developed in order to study bulk microarray data. And this model had an adaptive sparsity constraint that was introduced by having a gamma prior on every single matrix element that we learned from the factorization, and is a strictly non-negative model. So this is a Bayesian non-negative matrix factorization with a gamma prior. And what's unique about this is that we have the hyperparameter that we're learning from the data is on the shape parameter of the gamma distribution. So what does this do if we think of a gamma as a sum of exponentials? What this is going to do is tend to have our data be constrained towards being very sparse when our alpha value is low, and to being actually constrained away from sparsity when our alpha value is high. So we end up with a matrix factorization that's learning an adaptive sparsity that's tuned to the data set that it's applied to, which is particularly important for single cell data, as we know, which has so much dropout and so much sparsity. We really want to be able to model that within our data while accounting for genes where there might be real biological sources of signal that are constrained away from zero. And this model enables us to do that. So we've applied this model in a number of different contexts to look at cell state transitions. And Genevieve did a lot of her work benchmarking this algorithm in the context of developmental biology. And she and I always have a joke in my lab that she takes all of the developmental biology because it's super constrained, and we know that if we test it, if the algorithm doesn't work, it's not going to work anywhere. Whereas I take everything in cancer, and I'm like, this is great. All the parameters are broken down in cancer because the whole system is so much more stochastic. It gives me a parameter sensitivity, a biological parameter sensitivity to my algorithm to see where it works or it doesn't work. So what Emily Davis Marcisak in my lab did is she wanted to take this algorithm and see what happens if we take this approach that's been benchmarked in developmental biology and apply it to a case of immunotherapy in a mouse model to see, can we figure out the cell type heterogeneity in response to different immunotherapies in a mouse treated model. So she collected public domain single cell data from a paper by Gubin et al., where what Gubin et al. Did was they took single cell data from a control mouse, also treated with anti-PD-1, anti-CTLA-4, and the combination. So these are 2 different immunotherapies and their combination in a sarcoma model that's known to be sensitive to immunotherapy. And Gubin went on and did the standard sort of clustering analysis that we're used to seeing on UMAPs and associated different cellular abundances with the immunotherapy response. And so again, Emily wanted to go a little bit deeper and see, can we use the types of cell signatures that we learn that are independent of cell type in order to infer if there's other molecular processes that are underlying this response and resistance. So not surprisingly, she applied our COGAPS matrix factorization algorithm to these data And what she observed when this was a large number of patterns where some separated out the myeloid and lymphoid populations. And interesting, within the lymphoid populations, one of the patterns was observed to be enriched specifically in this sort of top peak of the duct, which if you dig in is a subset of the NK cells that have been annotated in this population. And not only is this not this, so the question is this technical, is this biological? What's interesting is comparing this across the different treatment groups. What Emily observed is that this was associated with NK cell activation uniquely in anti-CTLA-4 treated mice. So the question becomes, okay, so it's associated with anti-CTLA-4 treatment. And then what is it doing, right? So the advantage of matrix factorization is not only do we get which cells something's associated with, but we also get a corresponding set of gene weights, which enables us to label the molecular function that's associated with the patterns that are learned from the data. So Emily overlaid the gene weights onto pseudotime and found a strong association of our pattern with pseudotime in the NK cell population. And looking at the genes that were associated with the pattern weights from this, found that they were also associated with NK cell activation in the data. So the question is this NK cell activation in response to anti-CTLA-4 treatment a hat trick of this one mouse model? Or is it something that's actually relevant to human response to anti-CTLA-4, and that might be a mechanism of response over and above the T cell response that we should be considering as we're looking at mechanisms of response and resistance to therapy? So what we want to do is then take and see how much of what we've learned in the mouse is going to go on and be preserved in human data sets. And so what Genevieve had gone on to do with Gaurav Sharma is to say, okay, if I learn these patterns that are associated with different gene weights, and I've annotated them to different functions through matrix factorization, what I can then do is see how much the signatures that I've learned of matrix from the matrix factorization, for example, in the mouse, can project onto a new data set, for example, from human, so that I can overlay the types of responses that I see in one system and another and query where they're occurring and what the underlying biology of the system is. So again, Genevieve went on to beautifully demonstrate this for understanding the conservation of the developing retina between mouse and human. And we wanted to see if this could enable us to better track the human response to anti-CTLA-4 and whether there was any NK cell activation with it. So we took the Dugubin et al. Data as our training set for learning these features that I've shown you. And then we looked in TCGA and 2 single cell data sets of human metastatic melanoma treated with different immunotherapies. And what we observed is that when we projected this NK cell activation signature onto pre-treatment single cell RNA sequencing data from human metastatic melanoma, we observed that in anti-CTLA-4 treated NK cells, we observe an enrichment of this NK cell activation in the responders relative to the non-responders, suggesting that there is a role for this mechanism in human response to anti-CTLA-4 treatment. And again, it was uniquely occurring in the anti-CTLA-4 treated group as opposed to the PD-1, consistent with what we saw in our mouse model. So we also want to then go back and see, okay, is this correlative or is this indeed something functional? So working with Alison Fitzgerald and Lou Wiener's lab, what Emily went on to do is then see whether we could look at the expression of CTLA-4 in human NK cell lines to see indeed is CTLA-4 a mechanism that can be expressed in NK cells, since it's only thought to be a checkpoint in T cells. And she indeed found it in NK cell lines as well as human donor T cells through both QRT-PCR and Western blot. and then went on to do further staining and imaging and further experimental validation of the function. So this gives us one way of studying cellular activation and heterogeneity as it's associated with immunotherapy response, and really a means of doing preclinical bench to bedside research computationally, where we can start to learn features in a computational system from a mouse model and query them in humans, so we can start to get at some in silico mechanisms of understanding therapies that expand our arsenal to study human disease. So the other trick with immunotherapy is, if we think about these different cellular compositions of the tumors and their relative microenvironments with different immune cells, it's not only what cell types are there, it's how are they distributed spatially. So if I have a lot of immunosuppressive cells that are embedded inside my tumor, or I've activated T cells that are not able to infiltrate, or I've exhausted T cells that are infiltrating my tumor, all three of these are spatial constructions that would make you resistant to immunotherapy for different reasons. And particularly this middle case is one that you're going to lose by virtue of doing dissociation-based single-cell assays, because you might see a large number of activated T cells with your tumor, but if they're not able to infiltrate, they might not have any function on actually inducing a therapeutic response. So we're fortunate, and I think there's been a lot of work presented here and in other meetings, that these single-cell suspension-based technologies are all giving way, and there's a new explosion in spatial molecular technologies, both at the proteomics and transcriptomics and even multiomics level, that are really fully available to now start characterizing these systems. Each of these technologies, between spatial and, as people are well aware here, between spatial and suspension-based, have different pros and cons, as well as proteomics to RNA, that give you different pieces of information about the biological system that you may want to piece together to understand therapeutic response and resistance. So if we're thinking about these mechanisms of therapeutic resistance, how are we going to study them directly in patients? And how are we going to understand if we're giving therapies that are designed to induce immunotherapy sensitivity, how are we going to actually test the cellular and molecular pathways that are associated with that response in order to better map it and start seeing if our treatment combinations are really having the effect we hypothesize they are? So one common way of doing this now is through neoadjuvant clinical trials. And one example of this that we recently performed in collaboration with Mark Yarchwin was led by Wanjin Ho, where Mark had designed a neoadjuvant clinical trial in liver cancer. And so what they did is, liver cancer tends to be very immunosuppressive, so it's not a tumor that would naturally respond to immunotherapy. So what they did is give a lead-in with cabozantinib, designed to target the VEGF pathway and other pathways, and then add in nivolumab immunotherapy after that was done, with the idea that cabozantinib would rewire the tumor microenvironment to induce more immune cells to infiltrate, that then when you give nivolumab therapy enables them to activate and have clonal expansion. The unique thing about these neoadjuvant clinical trials is there's no prior therapy. And then after the therapeutics are given, the patients go in for surgery. So you're able to isolate uniquely the response to therapy and distinguish that between responders and non-responders, because you have these large chunks of tissue that are obtained from surgery where you've isolated the therapy that patients are obtaining so that you can get a case and control study. So in this study, not only was this, useful for me as a computational person to try to dissect the tumor microenvironment, far more importantly, this study had 5 of these tumors who were treated in this phase 1 clinical trial demonstrate a major or complete pathologic response to this immunotherapy treatment. It was really a phenomenal response to this. So again, we have these resections after treatment. So what Juan went forward and did is take research biospecimens of these tumor samples after treatment and apply imaging mass cytometry, the spatial proteomics, to see if we're able to understand the immune landscape, and did we indeed rewire the tumor immune microenvironment due to this combination therapy. So the trick with imaging mass cytometry is that it's limited to the number of markers that you put in. So you get about 30 or 40 protein markers. Juan, in this case, developed a beautiful panel that was really aimed at pan-immune and tailored to the liver, where we have myeloid markers, lymphoid markers, and hepatocyte markers, as well as some functional immune markers. I'm not going to talk too much about the technical artifacts in imaging mass cytometry data. I'm going to focus on the biology that we get out of it. But I will say this is a super ripe area for research in terms of batch effects between slides, image segmentation, normalization. You know, I think people are really focused on this in the single cell space, but I would make a huge call out that spatial proteomics is in desperate need of some computational thinking in terms of robust study design and analysis. So again, when you get these spatial proteomics data, you know, what you typically get are these beautiful images, which is where, you know, most of the work tends to stop unless, you know, you're more computationally minded. And even looking at these data visually, what you can see in the spatial proteomics, assuming you're not colorblind, and I apologize to anyone in the audience who is that the non-responders have enhanced green staining associated with E-cadherin, and this is associated with an increased density of tumor cells that are occurring in the sample. Whereas in the responders, you see lower E-cadherin staining, which is associated with less dense tumor cells that's counteracted by increase in CD16 and CD68. So you're seeing this increase of immune markers infiltrating while your tumor cells are shrinking out in the responders. So what do we do with spatial proteomics data? So we segment these into single cells. You get a protein expression by cell for these images. And then the typical thing that people do is apply a clustering-based algorithm, typically flow sum clustering in the case of IMC, in order to understand the different cell groups and quantify what cellular proportions are occurring within these samples, very similar to the types of analyses we do with single-cell RNA-seq. This gives us then a quantitative metric that we're able to compare how do the proportions of cell types change more quantitatively than showed in the imaging data. So Juan went on to apply this, and indeed, when we look at the data on the right, what we observe in the non-responders in the TMAs is this increased number of blue cells, which are the malignant hepatocytes, whereas in the responders, we observe more of these yellow and orange cells, which are immune killing cells that are coming in and infiltrating the tumor. So this gives us one quantitative metrics of cellular abundance, but the whole power of these technologies is we have the spatial information. So we want to know not only are there more immune cells, but are they actually more infiltrated within the tumor, and are they recognizing the tumor cells and more adjacent to them? So what Juan went on to do is compute the spatial distance between all of the clusters, one to another, find the average distance then between the cells using a minimal spanning tree, and define that minimal spanning tree to define what is the typical relationship and spatial relationship of cells one relative to another. And he did this separately for the samples that were responders and non-responders. And when you apply this analysis, what you observe is this orange group of the lymphocytes and neutrophils that are able to successfully mount an immune attack are segregated from these teal cells that are all the malignant hepatocytes. So those form in separate clusters and they're spatially segregated away from one another. Whereas if you look at the responders, there's a tendency to have your immune cells co-localized with your tumor cells, again, supporting this notion that you're having successful immune infiltration and immune killing. And I think I've seen a lot of posters and talks at this meeting, this ability of how do we really leverage the spatial information to understand these relationships across the whole slide and in different samples, and compute differences between groups, I think is just a wide open area for graph theory that I would love to see more folks working on. So the spatial proteomics is really powerful at giving us true single cell resolution with validated antibodies where we can really map this robust response visually and computationally. The challenge with it is that it's limited to what you put in. So you really need to know your markers going in order to get a good functional assay. And we might want to do additional discovery work as well to find unforeseen mechanisms that are relating to response and resistance, because, if we knew it all, we would have the therapies in place. So Luciani Kagahara from my group went on and with the same cohort of samples applied spatial transcriptomics profiling to these data. And for those not familiar with it, what spatial transcriptomics data provides is it provides an H&E combined with full transcriptomic profiling that's not quite at a single cell level. It's at like a 5-micron resolution at little spots that are throughout the entire slide. So she applied this in responders and non-responders and worked with our pathologists specifically to find ideal regions with both tumor and immune cells, with the caveat that in the responders, like we saw, there was a lot more limited tumor regions to choose from in terms of the analysis. You can apply the same type of clustering analysis to single cell transcript or to spatial transcriptomics as is applied in single cell analysis using Seurat or whatever pipeline, is sort of in place. And using this, what Luciani found was, again, this confirming the same sort of difference between responders and non-responders, where there was more tumor cells in the non-responders, and the responders had a shrinkage of tumor cells with more immune cells. And through the spatial transcriptomics, we were also able to observe densities of fibroblasts that we weren't able to observe in the panel with IMC. Interestingly, when we went into one of the responders and we looked at the tumor regions, and we just looked at markers of immune response, so for example, HLA-1 and HLA-2, and here I'm showing only HLA-1, what we observed in this tumor sample was that there was heterogeneity in these immune response genes on the tumor regions, suggesting that we were seeing antigen recognition and response only in the tumor region on the right, but not the one on the left. And so the question is, what is moderating that difference in the response? If we look at straight up clustering, we observe two dominant clusters between these again, where we see more immune infiltration occurring in this red cluster with the tumor regions defined by the blue cluster, whereas in this green region that's associated with the cluster, we're not able to segregate, but we see a mixture of tumor cells and fibroblasts. So we want to go a little bit deeper because this is leading us to hypothesize that the fibroblasts might be blocking this immune response, but it's difficult to tell what's going on when it's all mixed together through clustering. So not surprisingly, we're going to go back to our trusty old COGAPS method in order to start dissecting out different patterns of different immune cells and tumor cells within this data set. So Atul Deshpande and Melanie Loth and my group went on to then apply COGAPS to these data. And what they observed is that when they apply this matrix factorization, instead of having the fibroblasts co-localized with the tumor cells and having a distinct tumor cluster on the right and on the left, what they observe is that there's one common tumor pattern that spans all the tumor regions, this pattern 2, but there's another pattern that's very unique to this region on the left that we're hypothesizing is this immune resistance region. And we get a third pattern that is totally mapped to the immune cells in this sample. So the advantage of spatial transcriptomics data is that if we have these different patterns, what we can start to do is overlay them and ask, where are the regions at which they interact? Because presumably where we see these interacting regions are where we are going to have molecular changes that are associated with cell-to-cell interactions. So what Atul did is developed a kernel-based method that if you have distinct patterns, you could then quantify what are the precise regions in spatial transcriptomics where you see tumor and immune cells co-occurring. And then over and above that, what you can do is when you see regions where they're co-occurring, he developed an approach called SpaceMarkers to try to determine what are the genes that are changing due to that cellular co-localization, suggestive of direct interactions. So the way this method works is that you look at the data and you can ask, what are the genes that are expressed in one region that are associated with one cell type? What are the regions that are expressed with another? And if I'm in this region of cellular co-localization, is my gene expression change over and above what I would see if I just had cells expressed in one region or another? So using this SpaceMarkers approach, we applied it to these data. And I apologize, the interaction region might not be showing up well on the screen. But I assure you, we have different interaction regions with pattern 2 on the left than pattern 1 on the right. And when we apply this, what we observe is successful immune killing by immune infiltration in tumor pattern 2, whereas on the left, we observe fibroblasts blocking tumor immune interactions in pattern 1. Sadly, what this was leading us to hypothesize is that this patient, again, all these data are leading us to hypothesize that this patient has a remaining region of the tumor that's immunotherapy resistance induced through fibroblasts. And unfortunately, this was actually the first patient from the clinical trial to recur due to their cancer. And it's actually the first time I've ever gotten cancer-related genomics data where we started to analyze it and got concerned about the patient and then correctly predicted that response. Obviously, it's an N of 1, but it really does start to get us at something that's likely a mechanistic form of resistance directly in a patient. So if we're going to design the next therapy, what we need to do is go beyond saying, okay, they're fibroblasts, to seeing, can we figure out exactly which cell types are expressing these genes that are changing due to the molecular interactions, because we're going to want to target specific cell types. So our same transfer learning approach that we developed and used for mouse to human data, we can also apply from spatial to non-spatial data. And so we had matched single-cell RNA sequencing data with this tumor sample. And we were able to use our projection-based approach to find these distinct tumor patterns as well as the immune pattern in different subsets of the cells that we obtained from our single-cell data. And mapping the space markers onto these single-cell data, we're able to observe that a subset of the cells do express these markers that are co-enriched in both, enabling us to hypothesize that we're able to directly identify which cells these interactions are occurring in. So with that, I'm going to end my talk and say what we've learned is that cellular composition really is driving immunotherapy response and resistance. Single-cell multiomics are ideally suited to resolve the state-of-the-tumor microenvironment. COGAPS is the best, but seriously, there's a lot of great latent space analysis algorithms out there, and latent factor analyses, I think, are super important for these data sets, but making them in a way that's interpretable back to the biology. And finally, interaction statistics really enable us to model these tumor immune interactions. Of course, none of this would be possible without my great lab and funders. And I would encourage folks, we have a totally different topic about RNA forecasting that Ross and Urbe is going to give a talk on, one of my graduate students tomorrow in the machine learning seminar. So I'd really encourage people to go and listen to that for other work. And of course, I have to make the shameless plug, like every PI here. In our institute, we're starting a new postdoctoral fellows program that's really aimed at how do we get computational people more versed in some of the clinical translational problems so that the methodologies that we're developing and the analysis tools and analyses can be really translationally and therapeutically relevant. So we're building a new cohort-based postdoctoral program that's going to involve multiple computational and clinical and experimental investigators throughout the Hopkins Cancer Center. So if folks are interested in this, I'll be at the job fair on Thursday. But you guys can also find me, and I'd really encourage people to reach out, as well as several faculty openings. So thanks, and I'm happy to take questions.
I have a question actually about the matrix factorization. I understand the principle is you're taking a data set, mapping it to a space that's on the data set, right?
Mm-hmm.
I was wondering if you had any sort of current mechanisms for genes that are dominant that may influence protection when they express the high expression.
Yeah, we haven't seen that happen as much as you'd expect. Genevieve's been working, I believe she has a preprint out on developing a weight for how much each gene is weighted in the projection. So there's a preprint out on that. That's a new method that we've been working on, because you could imagine that having a role, and then also what's different in the projection can be relevant to biology as well. And yeah, so that we did in our developmental cell paper was to look at those differences through that approach. Oh, sorry, absolutely. Yeah. So the question is whether we've used cell phone DB or Giotto or ligand receptor sort of network style inference on the spatial transcriptomics data. So I didn't present that here today, but we have started to use those tools. And we've developed a third one called Domino in collaboration with Lana Garmeyer and Jennifer Alisieff that we've particularly been using on these data. And we found that they do work pretty well. The challenge with them is that they're not including the spatial information, which is what you want to get out. So a tool is now working on extending the SpaceMarkers approach to see if we can better consider ligand receptor interactions spatially. But I do think that's going to be a very emerging area. Yeah, so that's a great, so the question is about tuning. We have our Bayesian matrix factorization algorithm, and we have this adaptive sparsity that's tuned through shape parameter in our gamma prior. We have, one of the things is that that's the sparsity is learned as part of the sampling. So in addition to learning the matrix elements themselves, we also sample that sparsity. So it's driven by the data. But there's a trade-off in that between how sparse is the data and what's the magnitude of each matrix element, right? Because if you have fewer possible values of matrix elements, then they're going to tend to be larger. Whereas if it's more, then they tend to be smoother. And so we don't have a great heuristic that we've done this. I can tell you that I manually tuned every single value when I was developing this algorithm as a postdoc, and found that this was sort of what gave that our initial hyperparameter imbalance is what gave sort of the right balance between sparsity and biological processes. And we found that by looking at the smoothness of time course data solutions learned. But I think how you could do this in a fully unsupervised way, or fully sort of data-driven way, is going to be really important. And, I also think there's room for gradient-based or variational algorithms with similar constraints that would be a lot more efficient than our MCMC approach that we're using. But the trick is this is essentially a combination of an L0 and an L1 norm. And so it's a little bit tricky to implement in that. But I'd be happy to talk to anyone on the computer science side about that. Yeah, so the histology annotations in this data set, so the question is, where are the histology annotations coming from? So they are coming from, I will not collect spatial transcriptomics data if we don't have a pathologist on board, because I want to make sure that we're actually getting a meaningful region of the tumor. So they're very much part of the analysis team and part of what we do. And so we've worked very carefully on that, and they go through and annotate. We have a paper that we're probably going to preprint this week. So we just submitted it and should be out any day now, where we also took in, so in the frozen case, it's a little bit tricky because most pathologists are used to looking at H&E on FFPE sections. And so it's actually kind of difficult for our pathologists to annotate our cells in the frozen sections. And so one of the advantages we've seen of the new FFPE technologies for spatial transcriptomics is that the H&E is easier for our pathologists to work with. And one of our collaborators, Denny Wirtz, and Ashley Keeman from his group, had developed an automated cell type annotator from H&E in the case of the pancreas. And so we were able to use that on top of FFPE data from spatial transcriptomics. And that by having the automated cell type annotation from the H&E and integrating it, we were actually able to regress out so we could get single cell level resolution from that. But I didn't present that today. Yeah. Yeah, another great question of, are we taking multiple slices and looking in 3D, or are we looking at one slice in 2D? In this case, this was one slice in 2D that I presented here today. All the work we've done so far has been 2D. We just got a new center grant, again, with Denny Wirtz, to look at 3D on multiple sections, and we're going to start digging into that in pancreas, so stay tuned. But they've done beautiful work in H&E and shown, ooh. Sean, whoa.